This video gives you a quick overview of respiration for the AQA A-level biology spec. I will be going through an overall summary of respiration, and then we will be looking at particularly glycolysis and then the link reaction. I'll go through the main points you're going to need to know. There are some extra things that you could learn, but I think it's best with respiration to stick to the points. And then please do look out for any later videos that I'm going to be looking at exam technique. So a quick overview. Respiration is a process that allows cells to produce ATP from glucose, and it can occur in the presence of oxygen or without oxygen. So with oxygen is aerobically and without oxygen is anaerobically. When you studied biological molecules, you would have gone through ATP, but this is just a quick recap. So ATP forms when chemical reactions release sufficient energy to bond a third phosphate group to ADP. Notice you can use the words ATP and ADP. You don't need to use the full names in an answer. So just very quickly, ATP is an energy currency for the cell. It mainly stores the energy for the cell and releases it when it's needed. So you can see that you get a reaction between ATP, and then when the phosphate is removed, energy is released. This reaction is reversible. It's catalyzed as well by ATPase. ATP breaks down when there is a demand for energy, so where there's high metabolism in such things as your muscles. So aerobic respiration, it occurs in both the cytoplasm and the mitochondria. You'll need to remember the basic structure of the mitochondria. You would have done this when you looked at cell biology and cell ultrastructure earlier in the course, and you really do need to be able to label them. Particularly of importance is the matrix, and then the inner membrane and the outer membrane, and the folds that increase the surface area known as the cristae or cristae. Before we get started, just a reminder again of GCSE chemistry related to redox or redox reactions. So it's when something is reduced or oxidized, and this happens a lot in the cycles that we're going to look at soon. So again, when something is reduced, it may have gained electrons, gained hydrogen, and lost oxygen. And as it says here, oxidation of one molecule always involves reduction of another. So when something is oxidized, it then will have lost electrons, lost hydrogen, or gained oxygen. And you might remember the acronym oil rig. So oxidation is loss, reduction is gain. It's just a helpful acronym just to describe the term given to a loss or gain of electrons. Something to remember. So let's get on and look at the different aspects of respiration. So as you can see by this image, there are four main parts to aerobic respiration, and that's what we're going to talk about first. And the first one is glycolysis, second is the link reaction, then the Krebs cycle, and then the electron transport chain known as oxidative phosphorylation. The first three stages are a series of reactions, so glycolysis, link reaction, Krebs cycle. Some of those reactions, there will be a small amount of ATP produced, but the products from these first three stages are used to produce ATP in the electron transport chain. So the first stage is glycolysis, and both aerobic and anaerobic respiration occurs here. It happens in the cytoplasm because glucose is too large to pass through the outer membrane of the mitochondria. So glycolysis involves the breakdown of one molecule of glucose, which is a 6-carbon. So if you remember, it is C6H12O6, known as a 6-carbon molecule, and it forms two molecules of something called pyruvate, which is a 3-carbon. We need to talk about the two stages. The first stage, phosphorylation, which means adding a phosphate group, and the second one, oxidation, which is the loss of hydrogen electrons and the gain of oxygen. So it's important to state that one glucose molecule forms two molecules of pyruvate, and that will be important as we get to the next stages. So before glucose can be split into two, glucose needs to be made more reactive, and that's by the addition of two phosphate molecules. So the addition of those two phosphate molecules is known as phosphorylation. So the phosphate molecules, they are coming from the ATP. Okay, you can see ATP being used there, and the hydrolysis of two ATP molecules, we got them 1 and 2 there, okay, and to ADP. So this provides energy to activate the glucose, lowers the activation energy for some enzyme controlled reactions to occur, and then luckily you don't need to know that molecule, you just need to know that phosphorylation occurs and it splits into two 3-carbon molecules known as triose phosphate. And after two triose phosphates are produced, it's then oxidized, which means it loses hydrogen and it forms two molecules of pyruvate. So let's talk about the particular steps there, because there's quite a bit going on there, as you can see in the diagram. Now, in glycolysis, it requires the help of a coenzyme. A coenzyme is not an enzyme, but it allows enzymes to work more efficiently. So NAD, in this case, is the coenzyme, and as the triose phosphate is oxidized and it loses the hydrogen, that is then combined to make reduced NAD. That will be then used in the electron transport chain at the end of the reaction. So NAD helps the enzymes that catalyze the oxidation reactions by accepting and carrying the hydrogen atoms that have been removed. Now, in terms of the triose phosphate contains a phosphate, and that is used, it combined with ADP, and it produces ATP. Now, as you can see, there's two molecules of triose phosphate. Each molecule, it makes or produces two ATP molecules each. So therefore, we can say there is a net gain of 2 ATP. The reason there is a net gain is that we use 2 ATP in the energy to split the glucose, but we gain 4, so it's a net gain of 2 ATP. So to summarize the final things, NAD collects the hydrogen ions from when the triose phosphate is converted into pyruvate as it's oxidized, and that forms 2 reduced NAD or NADH, and then 4 ATP are produced, but 2 are used up in stage 1, so there's a net gain of 2 ATP. So again, just to recap, in glycolysis there is a net gain of 2 ATP, the production of 2 reduced NAD that will go on to the electron transport chain, and finally at the end of glycolysis 2 molecules of pyruvate. So from glucose we now have 2 3-carbon pyruvates ready to go into the next stage, which is the link reaction. Okay, so we have our ×2 of pyruvate, and now it is small enough to gain access into the matrix, the mitochondrial matrix, for the next stages of respiration, which happens to be the link reaction. Now the pyruvate is actively transported across the mitochondrial envelope and into the matrix by transport proteins. So There is active transport going on here, which is worth remembering if you get an essay about transport across membranes, because this is quite a good example. So this second stage, the link reaction, its purpose is to convert the 3-carbon pyruvate produced in glycolysis into acetyl coenzyme A, or acetyl-CoA. It's known as CoA as well. This is only now in aerobic respiration, not anaerobic respiration. So pyruvate, a 3-carbon molecule. The first thing to happen is a carboxyl group is removed from pyruvate, which is known as decarboxylation. So 1 carbon is removed, and this forms carbon dioxide. So this is where some of the carbon dioxide is created when we breathe out, or when it will leave plants after respiration. In that process, NAD, there is reduced NAD produced, and then you have a substance called acetate, a 2-carbon acetate. So with the reduced NAD, that's because at the same time pyruvate is oxidized, and so therefore it forms the acetate. NAD is reduced to form NADH. And finally, so acetate is combined with the coenzyme A, and that forms acetyl coenzyme A. No ATP is produced in this reaction. So just to recap, related to the link reaction, you need to remember that actually there are 2 pyruvates, okay, molecules for every glucose molecule. So this link reaction will happen twice. Okay, so this happens twice. And that's important to remember. So the products of the link reaction that we're going to talk about in a minute go to the Krebs cycle and the electron transport chain. So for each 2 pyruvates, we're going to have the following. So we're going to have 2 acetyl coenzyme A, which will go into the Krebs cycle, which is on my next video. We then have 2 carbon dioxide, those will be released as a waste product, and then finally 2 reduced NAD, which those will go to the electron transport chain. So that brings us to the end of looking at glycolysis and the link reaction. My next video will go into the Krebs cycle, and then I intend to do a final video on the ETC, and then we'll be looking at some exam questions. Hope you found this useful. If you have, please remember to subscribe, as there'll be more A-level biology videos coming soon. So happy revising, and I hope you found it useful.